So I'm gonna sort of try in the next sort of 3/4 of an hour or so to actually go through basically, I guess, a background and a kind of primer to sort of some of the things you'll learn a bit more about later on, but kind of give you an idea of sort of how genome engineering actually sort of started off and kind of where we're at now and what we can do with it and the different things that are available. So I don't know, many of you may know, but actually sort of the big sort of splash in CRISPR came in sort of early 2013, so just over 10 years ago now, when these four papers were timed to come out together. Um, and they basically demonstrated the application of this CRISPR-Cas9 system from a bacterial defense system into either human cells or zebrafish. And since that point, I've kind of, up to a couple of years ago anyway, I kind of kept a track on the number of citations in PubMed. And by sort of, it's actually leveled off a bit now, but it's sort of still about the same as this level. So there's about 1% of all PubMed citations that actually contain CRISPR or Cas9 in the title from 2022. And that's pretty similar to date. So this, I think, is testament to how well it worked and how sort of applicable it is across the whole of biomedical research, really. And it's now become a sort of generalizable tool that actually has been very, very valuable for genetic analysis in both what would be considered genetic model systems, where it's made that process much easier, but also allowed us to use non-genetic model systems such as human cells, and everything else, everything from sort of pigs to primates, to plants and everything in between. But I think it's also worth just highlighting, and for you all to know, I think you probably know this as well, that this has now sort of developed into even therapeutics. And this was a very big sort of scandal a few years ago from this Chinese scientist who actually sort of edited in the germline of a human baby. So very, very early, they injected all the CRISPR reagents into that embryo. And this actually resulted in him going into prison. He's now out of prison, but and there was a big outcry and a lot of sort of clamping down on this in the worldwide literature. But I think it's worth highlighting maybe because you guys in the room will be the ones that kind of have to explain this to the general population. So I think it's well worthwhile you being aware of some of the things that are going on, and it's important to have a kind of scientifically valid viewpoint on these kind of things. But also, it's been incredibly successful, and I think I'm just going to skip to the data here. This is actually one of the first sort of therapeutics that's actually been adopted now, and this is actually for treatment of sickle cell anemia and beta thalassemia. And I think this is very, very striking. So this is not a germline therapy. This is actually a somatic therapy. And you can see here, this is the set of patients that were treated with this. These little purple dots here are what are called vaso-occlusive crises, which are basically where blood vessels get clogged and they're very painful and very difficult. So you can see over this couple of years, there's a lot of all of these patients have a certain number of these. Then at this point in time, they gave these a CRISPR therapeutic that actually is targeting a regulatory element in this case, actually for deletion, and that upregulates the fetal hemoglobin, which means that it can rescue the defects that arise because of the sickle cell mutation. And after that point, you can see the number of purple triangles basically drops to zero. So this is, I think, incredibly striking and a really nice proof of principle that this is actually will work in humans. So not only as a kind of research tool, but I think this is kind of a demonstration that actually this is something that could be useful as a therapy as well. And this happened really very quickly. If you think about it, it was sort of from 2013 when the CRISPR sort of technology was applied first to human cells, showed that this worked in cell lines in sort of 2015. And by sort of 2023, this has actually been approved in the UK as a therapy.
What does the green and blue and orange mean on that
So the
chart?
green is just how long they've actually looked at them for. And I think probably the blue and the orange are kind of the two treatments that they were given. So when they gave the first one and then when they gave the second one.
How much is this therapy?
I've no idea. Quite expensive, I would imagine, because this involves taking basically HSCs from a patient, editing them in vitro, and then putting them back in as a bone marrow transplant. So I would imagine this is a very, very expensive therapy, but it is a
And
one-off treatment. You only have to do it once. So that's, I guess, just a bit of an intro into kind of, where things started and where they're at now. And I'm going to give you a bit of background on sort of, what it actually is, CRISPR, which many of you will know, but hopefully you'll learn something new. Um, Sort of some of the basic sort of things that you can do with it, like sort of in-cell mutagenesis and gene targeting, but then also go into some of the more exciting, sort of more recent applications of this technology as well, just to give you a flavor of what those might be. I don't know if many of you know, but actually, sort of CRISPR was discovered as actually from these regularly interspersed short palindromic repeats in bacterial genomes. So these are actually a palindromic repeat of a sequence, which actually turns out to be the backbone of the CRISPR RNA, with little sequences in between, which actually turn out to be derived from viruses. And this was discovered way back in 1987, and nobody really knew what they were, but it's sort of in the kind of early 2010s, this sort of discovered to be transcribed into and processed into these little CRISPR RNAs, which consist of this repeat region and then the viral sequence. And it turns out that this is actually a viral defense mechanism in bacteria. So it was actually discovered completely serendipitously, really, from studying sort of yogurt bacteria and why they didn't get sort of phage infections. And what it turns out is that this little sequence of viral DNA is kind of taken from the virus when it invades a cell and is sort of stuck into this array in the genome. And then that sequence is used as a sequence specific guide for a nuclease, which in this case is Cas9, that goes and cuts the viral DNA if it invades again into the cell. So this is like an adaptive immune system for bacteria. What it turns out is that actually this then becomes this complex, which has this what would have originally been a viral sequence and this kind of backbone sequence, along with this protein that's a sequence-specific endonuclease. This actually is targeted to its target site by simple base pairing rules between the 20 nucleotide sequence in the guide RNA and the sequence of the genome. Now, you can change that sequence for whatever sequence you want it to be. And this was the realization of these couple of papers in 2012, where they realized that actually just by changing this 20 nucleotide sequence, you could actually reprogram the nuclease to cut pretty much anywhere you like in the genome with very predictable rules. So it was different to most of the protein-based targeting that was used up until that point, like talons and zinc fingers, where actually the rules behind the protein DNA interaction were quite difficult to predict. In this case, it's just base pairing, so it's very, very simple to predict. The only constraint on this is this protospacer adjacent motif, which doesn't appear in the guide RNA, so it's often a mistake that people make. This is actually recognized by the Cas9 protein in the context of the DNA. And this, if you like, helps to, it's originally there to prevent it from cutting that sort of repeat region within the genome. But actually, it does provide a little bit of constraint on where you can actually target, but not terribly, because this should happen every 8 base pairs on average in a random genome. This means that actually by changing these 20 nucleotides, you can get it to target anywhere. And those papers in 2013 showed that actually you can transition this whole machinery into a human cell or a zebrafish, and actually it will still work, which I still find quite remarkable when you consider that the human genome is so big and packaged in histones and things, but it does. And I think they were lucky in picking this particular example as well, because it works extremely well. So what often people sort of show CRISPR as is this very fine sort of tool for making delicate manipulations of the genome. But really what you're doing here is actually using the Cas9 protein to make a double strand break in the genome, which is this extremely sort of toxic lesion. So you're kind of smashing up that bit of DNA and then asking the cell to repair itself. And this is actually what we take advantage of when we're trying to make mutations, is we can create these double strand breaks literally anywhere we like in the genome. And then we utilize the repair processes that tend to go on in the cell. So these broadly fall into two categories. One is this non-homologous end joining, where the ends are sort of glued back together. And most of the time, this is fairly precise. But occasionally, if you keep making cuts and cuts and cuts, eventually it'll make a mistake. And that normally results in sort of small insertions and deletions around that cut site. And this is predominantly the form of repair in most cells, with the exception of a few, like mouse ES cells, which is why they were often used for gene targeting in the past, chicken DT40 and yeast and that kind of thing. The second repair is actually where you use a template, which endogenously would be the sister chromatid, so it would only happen after DNA replication. Um, and that uses that template as a way of being able to make a very precise repair. But obviously, you can take advantage of this as well by providing a template in vast excess of whatever desired sequence you like, and that will allow you to make a very delicate change. So that in normal cells would be called homologous combination. In the case of CRISPR, it's often called homology-directed repair because you're delivering a template of a desired sequence. So these are variable. I've put these kind of rough percentages on here, but that's, as I say, very highly system and cell type specific. So that will depend on the cells that you're delivering this into. So what can you do with that? So you can imagine that these kind of short insertions and deletions that you would get, which this is a sort of very old experiment that I did in flies, you can see there's kind of these few nucleotide insertions or deletions around the CRISPR cut site. These actually you can use for a variety of different purposes, like sort of making you can target in the middle of a protein coding sequence and introduce a frame shift that deletes the protein. That's the one that's used the most often. But also you can potentially target other small regulatory elements within the genome. You can actually put two of these cuts in, and some of the time it will ligate these two together or these two together, but sometimes it will also ligate the two black ones together, and that allows you to get sort of long deletions, sometimes even inversions of that intervening sequence if you're lucky, although that's less frequent. And as I say, most of the time people use this to make gene knockouts, so targeting protein coding sequence. But we've used it now for pretty much all of these different sort of other regulatory elements that you can see here. So, for instance, microRNA target sites in 3' UTRs, splice sites to affect sort of different splice variants, trying to remove specific promoters, trying to remove specific sort of transcription factor binding sites, that kind of thing. Anything with a small regulatory element or small sort of element that would be disrupted. And I think this is again a very old experiment from me, nearly over 10 years old now. But this is actually just, I think, to illustrate the heterogeneity in these. So this is a semi-random process. So when you take a plane of cells, it's actually Drosophila cell line, where you transfect in then a gene targeting the AGO1 gene. Sorry, a CRISPR targeting the AGO1 gene. And AGO1 in here is stained with red, so you can see it is a in the cytoplasm. It sits around the nucleus. When you introduce the CRISPR, you end up with this kind of situation where you see a lot of the cells have lost the protein, so you've actually removed the staining, but some of them still do, and this is for a number of reasons. It's things like in this case you've got sort of two or three different alleles within the cell, so actually you have to hit all three alleles. Some of these deletions will randomly be multiples of three, so they'll actually maintain the protein reading frame, and therefore your antibody might still stain, even though the antibody or the protein may or may not be functional. So this is inherent to the sort of processes that you end up with this very heterogeneous mix of different alleles, different mutations. And often a lot of the time that we spend is actually taking out individual cells, putting them into a dish, growing them up and analyzing those and genotyping them to make sure they are what we think they are. So actually, the process of creating the mutations is often the easy bit. It's actually the process of sort of finding out the cells which are mutated that is the more difficult one. So that's, I guess, the easiest one. And as I mentioned, that's kind of the primary target of a primary method of repair of these double strand breaks. But you can also sort of you can imagine that this other form of repair, this kind of HDR based repair, is actually very useful for doing things like GFP tagging or very delicate point mutations in the genome. And so quite a bit of our efforts have gone into kind of trying to bias the repair processes in this direction. And this can be used for tagging. It can be used to introduce point mutations, and actually sort of I'll mention in a bit how you might be able to do that in multiplex fashion so that you can tile across different point mutations, but also anything else that you can imagine. You know, for instance, we've used it to insert these recombinase sites that then mean that we can insert libraries of different variants into those regions. But one of the things that we work on quite a lot, and a number of other groups around the world do, is combining this with iPS cells. So for those of you that may not have come across them, so these are stem cells that are reprogrammed from somatic cells that are taken from a person, often fibroblasts or blood. And by introducing this combination of four transcription factors, you can drive them back into what is basically an embryonic stem cell-like state. These can then be propagated and kept and then differentiated into whatever disease relevant cells you'd like. So when we're talking about sort of neurodegeneration, for instance, which is something that we work on a lot, it's very difficult to chop open somebody's brain when they're alive and scoop out a bit of brain. That people don't tend to like that very much. So this is a way that we can get brain cells from those people that are genetically identical to the original patient, but have been produced solely in a dish. And this is a limitless supply as well, because as I say, these can be expanded pretty much indefinitely. Now, often what people have done is actually then taken cells from patients with a disease, from control patients which don't have a disease, do these differentiations and then compare between the two cells. Now, because each individual has a lot of mutations in their genome, this means that there's a good degree of variability between what those two cells look like. And importantly, you don't actually know that what the causal mutation is that is driving those differences, because there's often many millions between two individuals. So what we can do here is actually take either our patient-specific cells and correct the mutation that we think is causal, or take a wild type cell and introduce the mutation that we think is causal. And this means that then we can generate what we call an isogenic pair of cells, which differ in as little as one base in their genome. And then we can compare those two cells, and they're a lot more similar, as you can appreciate. And that means that you can actually see more subtle changes. And importantly, it allows you to establish that true causative sort of mutation that's actually driving the disease state, rather than inferring that from kind of a larger number of patients. So the way we do this is actually by introducing our Cas9. We actually do this now as a protein, which has a number of advantages in terms of longevity of expression. Is very short but very extreme. So we end up with a very sort of high spike of activity. Secondly, it's very easy. We just buy it up from a company. We actually have the guide RNA synthesized as a synthetic RNA as well. And then as a homology template for homology directed repair to make these point mutations, we use a single-stranded DNA oligo, which we just buy in from a company. So we mix those things together, we zap those into cells using this AMAXA, so nucleofection system, electroporation type system. And you can end up with a population where there's many of these edited single nucleotide changes, and also as a byproduct you get a lot of insertion and deletions as well. So then you can pick out individual cells and grow them up. And we actually had this with some guys in the cellular informatics team. We actually developed this way of being able to genotype using MySeq and actually visualize which cell clones are homozygous HDR, for instance, indel homozygotes, wild types, like this one, without actually having to go through a massive amount of effort to work it out. And we've done a heavy amount of optimization on this. Again, if you're interested, have a look at this paper, where by using various different inhibitors and improving culture conditions, we could get, in the best cases, up to kind of 70% of the desired single nucleotide change that we'd like to put in.
What does the axis mean on that plot?
This is actually a reporter system where we have a BFP gene, and when we create the cut with the CRISPR, if we get any kind of indels, we lose BFP fluorescence, which gives us this lower population. And if we introduce two point mutations by HDR, we shift the blue to green. So you can see that you actually get green fluorescence. So this is a measure of the HDR events, and this is the measure of the indel events. And this is cells which haven't taken up the protein. And actually, this is almost 100% delivery without any kind of selection.
Andrew, can we get the DNA PKI commercially?
Yes, yeah. No, it's all published, and it's all readily available.
Is it better than the HDR enhancer, the IDT cells?
Yes, it is. Yeah. So we can physically go through this process, and I think this is just to highlight. This is actually an example that we did in a gene called PTK2B, where it was quite successful. And actually, what we've done here is actually differentiated these cells with this single nucleotide change into microglia, and then we've compared the wild type and the mutant. And you can see here on the RNA-seq, these two principal components on PC2 pretty much separates by genotype, fortunately. So this tells us that actually the changes that we're looking at here are actually driven by the genotype. But this involved a lot of repeats of both the differentiation and the individual clones. But you can learn a lot of information from this. So this we can see is relevant to biological processes which are relevant to the disease, in this case, Alzheimer's disease. And also you can see there's kind of changes in chemokine release, which correspond to the changes in chemokine sort of expression that we see in the RNA-seq. So this, I think, is a nice example of where you can use these isogenic cell pairs to actually be able to extract useful information about what that mutation is causing in the cells. However, there's a couple of caveats to this whole process. So firstly, this is something that people don't often tell you. The efficiency is highly variable. So this is actually, again, some quite old data. But you can see here, all of these are different CRISPRs and target sites aiming to introduce a single nucleotide change. The total height of the bar is the mutagenic efficiency. And the blue bar is actually the HDR efficiency, so the introduction of single nucleotide changes. So this is quite old data, so things have improved a bit since here, so you can push everything up a little bit. But you can see that, first of all, the overall efficiency of mutagenesis is highly variable between guides. And secondly, the rates of HDR are also extremely variable, and in some cases almost zero. I should say these have all been pre-filtered for having some kind of guide activity as well. You tend to get maybe 20% of guides which don't work at all. So yeah, in the best cases, it's very easy. You can find, sort of 40 to 80% HDR, and you can get very nice results. But in the worst cases, it's almost impossible to find your particular clone of interest. So I think it's worth bearing this in mind when you're doing these experiments, is that it's not always equal, and it's worth testing out various different guide and HDR pairs to actually sort of maximize your chances of success.
Did you not use Cas9 fused with cell cycle regulated protein like Gemin? And I don't know if you see this.
Yeah, no, we've tried a huge number of these different permutations, and in our hands, they don't make any difference at all, really. So in some cases, we did actually try a couple of different fusions before that paper was published. One of which was the Geminin Degron, and there was also we included another one which was actually a dominant negative to one of the cell cycle repair proteins. Some of those have mild effects, but actually, when you're delivering with RMP into iPS cells, basically they don't make very big difference at all. So direct coupling of your template, putting in. So when we deliver them as plasmids, we can see differences, but that's because the general overall efficiency is much lower. So actually, probably it's something to do with the way in which we're delivering, the way in which we're growing the cells, which is already heavily optimized to mean that they're dividing as fast as they possibly can. So it's not to say they won't have an effect in certain situations, but in our hands they don't improve the efficiency in our case. And the only things that we found to make a difference are two. One is the sort of DNA PK inhibitor, so an NHEJ inhibitor. And again, there's an AstraZeneca compound that is freely available that is by far the best of those. Also a cold shock, which seems to also affect microhomology mediated repair. And that's just 32 degrees for 24 hours after nucleofection. And those two are additive. The cold shock has much less of an effect, but we are looking at sort of microhomology inhibitors as well, but we haven't got very far down that line. So combinations of DNA PK and microhomology inhibitors.
I have one question. These things you're doing in actively dividing cells.
Yeah.
For post-mitotic cells, what's the efficiency, and is there anything that you're doing to improve them?
It depends what post-mitotic cells you're talking
The
about.
neurons.
So neurons, HDR won't work because there's no G2. So you're not going to be able to get HDR to work at all. And HEJ works fine. I think there is some difference in toxicity there, which means that there's a slight difference in the selection, and also the delivery is difficult as well.
Base editing for the neurons.
We haven't tried. It should work to some extent. As I'll mention a bit later on, there is sort of, with base editing, it is a little bit, obviously the repair mechanisms are different. So it depends on whether those repair mechanisms actually exist within a neuron. But I think it should work. One of the constraints of that is also the kind of editing of multiple bases, either within the window or even slightly outside the window as well, which limits where you can use it a bit. But if you can use it, then it should be doable. But again, it's very, very, very variable in efficiency between sites. So you may be lucky. So yeah, what I've shown you is kind of a sort of a long-winded process. So this actually involves nucleofecting, picking colonies, growing them up, genotyping them by high throughput sequencing, freezing them down, thawing out the appropriate clones, regenotyping to check, growing those up, differentiating in multiple batches. So it's an enormous amount of effort to do just that single point mutation. And also there's a very big variability in a lot of these steps. So one is the variability between individual single cell clones. So you can see here these red ones are 3 individual wild type clones, and you can see they behave quite differently to each other. And also variabilities in differentiation, and you can see here that actually the vast bulk of the variance here is actually from differentiation to differentiation variability. So there's a lot of variability, and that means again you have to ramp up numbers. So this is a process that can work, but it's not necessarily very feasible for doing large numbers of these edits. So a lot of what we do now is now based on pooled screening, and I'll go into sort of some of the basics of that, but then also sort of some of the more funky versions of this as well. So when people are talking about screening, they generally talk about 2 types. I think arrayed screening is much more easy to understand. It's kind of basically you have a well where you perturb a particular gene in it, and then you have an assay that you're measuring your effect of that well. What we tend to focus on is these pooled screens, where instead of separating things out into wells, you actually generate a mix of cells where each cell within a population has a different mutation within it, and then you use some kind of single cell assay, be it growth, FACS, single cell RNA-seq, etc., to actually then deconvolve what the phenotype of that particular mutation is. And this is very nice for an individual lab because it means that you can do very large screens with relatively little outlay in equipment. This is actually sort of just growing cells, big amounts of cells in a dish. The principle behind this has been used quite a lot now, and the idea is that because those little sequences of 20 nucleotides that guide where the CRISPR cuts, they're very easy to synthesize on an oligo array in a similar way to sort of DNA sort of microarray sort of synthesis, and that means that you can create libraries of sequences that target up to every gene in the genome. And these were some of the early papers that then cloned those into a viral vector such that you could deliver these so that each cell contained one guide. And these have been incredibly powerful sort of across a lot of different applications to be able to do very large scale screens. Please don't go away and sort of use the libraries that were developed in these early papers. There's been a lot of development of these sort of since, and if you want this add gene page is actually a very good sort of reservoir of information about the most recent forms of the libraries that are much better ones to use than these original ones. But I think it's a very powerful way of being able to do genome-wide genetic screens for phenotype in human cells, for instance. And the principle behind this is that you deliver your library of vectors, each of which has a sort of guide that targets a different gene in the genome, into cells, normally using a virus, such that each cell takes up one virus. So you normally deliver it at low viral titer, so that you end up with only about a third of the cells taking up a virus, but each one then has one and only one. Then you select those cells that have taken up the virus using a drug selection or a FACS. And then you have a population where each cell is because these are integrating viruses, each cell is labeled with the guide as a measure of what gene is knocked out within it, and that guide will target that particular gene for a knockout. So you then have this mixed population within a dish, which you can then apply to various different stimuli. And the simplest one is just to grow them. So you can imagine if this orange cell knocked out a tumor suppressor gene, it might grow more and therefore overproliferate. If this purple one sort of caused sort of a cytostatic response, then that cell would stay constant for a while and then eventually die out because all of the other cells around it are growing out of the growing up, but it's not able to grow anymore. So what you can do is over time you can actually sequence these guide RNAs as a proxy of what the genotype of that cell is and follow their abundance over time. And if you do this by high throughput sequencing, you can do it for the whole genome. And therefore, you can identify cells that overproliferate over time or cells that drop off over time to give you a list of essential genes or genes that are tumor suppressor or sort of affecting cell growth. And actually, this is now being done across pretty much most of the standard 2D cancer cell lines in the world by this Cancer DepMap project. And this has been run partly out of Sanger, partly out of the Broad. And they've now, I think, screened, this is probably an underestimate, this is a while ago, but probably getting on for 1,000 different cancer cell lines across the whole genome in terms of knockouts. So we have a very good idea of how well these work, and these have been immensely powerful for identifying key genes that are involved in or that would be targets of cancer therapy. So they might affect particular types of cells growth, so particular types of cancer growth. So actually what we do a lot of work on is actually trying to push that to sort of more complex mutations, for instance. So we've been working quite closely with Matthew Garnett's lab here over the last few years, and we developed this system where we can actually deliver two guides into the same cell, separated by a tRNA that then gets processed out by endogenous RNases within the cell to separate out those two guides. And this allows us to look at genetic interactions. So we can use this to target two genes on one vector. And this is just an example from our preprint, which you can go and have a look at, where if we take two paralogous genes, so this is ASF1A and 1B, we knock them out individually, we don't see any change in growth phenotype. But if we knock them out together, we actually see a reduction in growth of the cells. So this is just, I guess, a proof of principle to show that can work, and then you can use that to study genetic interactions, which is incredibly powerful for cancer therapy, but also for a lot of other applications as well.
Can we do that with Cas12 multiplexing?
Yes. Again, I'll mention that briefly later on, but the problem with Cas12 is that the efficiency is generally a little bit less. And also, so often people get around that by putting in two guides per gene rather than one. And therefore, it's four guides per gene for a combinatorial screen. And that then means that there's a bit more toxicity of the double strand breaks. Also, you don't have all the Cas9 lines ready-made, which we have a lot of. So actually to remake all of the Cas12 or new Cas12 lines is a bit of a faff. So you're right, you can do. I think they're both valid strategies. I think this is something that we're more comfortable with. We also have a lot more information about the efficiency of the guides with Cas9 than we do about Cas12. And as I mentioned, even with the best predictions that you can get, you still can't predict desperately effectively in terms of overall efficiency. So I think that's sort of why we've gone in this way. We also set this up a few years ago, so it's before that really came about. Then you can also think about sort of looking in more complex cells. So this is actually from a colleague, Lewis, this is actually now just been accepted for publication, where they're actually doing, he's doing whole genome screens in iPS-derived neurons. And this is by no means a trivial exercise. You need an awful lot of cells to start with. But actually what he's done here is actually introduced the CRISPR editing at the neural precursor stage, and then fed the cells with fluorescently labeled tau. Then he can sort out the cells that no longer phagocytose the tau by fact sorting the fluorescent negative population, and then identify the genes by sequencing that and the input. And this gratifyingly comes up with LRP1 as one of the top hits, which is a known tau receptor, but also some interesting other genes here. LRRK2 is the one that he's particularly followed up on. This is actually sort of gives you a lot of extra information about the processes involved and the kind of the biology that's involved here. So it's a way of discovering new biology that you wouldn't necessarily have predicted from the literature. Another thing that we're doing quite a lot of, or have done quite a lot of now, is applying it to single cell readouts. So I mentioned that you can use pretty much any single cell readout that you can think about. Single cell RNA-seq is an obvious one of those. And this is becoming more accessible. It's still quite an expensive process, but it's definitely becoming cheaper and cheaper to do. And the principle here is that basically you can use your favorite single cell transcriptomic device to actually get whole genome or whole transcriptome mRNA expression. But if you can also capture the guide RNA, because this is an expressed RNA in the cell, it's not normally polyadenylated, but there's various tricks you can use to capture that in your single cell data. You can actually label each cell with the guide RNA it's expressing, and therefore presumably what knockout it might actually have within it. So this means then that you can actually understand the effects of every single gene knockout on the whole transcriptome. And this is again from a paper a few years ago now that we published, where in this case we've actually overexpressed using guide RNAs. But you can see each of these different clusters of cells is a different overexpressed gene from this list here, and you can see they separate transcriptomically quite well in this case. So this means that actually from this one experiment we can actually see this selection of about 30 gene overexpressions and their effects on the whole transcriptome all in one experiment. So it's an incredibly powerful tool, I think, to understand what's going on. Another thing that's going on very sort of a lot at Sanger is not just looking at knockout mutations, but also looking at point mutations. And one of the methods which we're using quite a lot is this saturation genome editing method, where actually you use multiplexed HDR. So in this case, instead of introducing indels, you actually introduce point mutations by HDR within an exon, and therefore you can actually introduce hundreds of those point mutations all at the same time and then follow their effects on growth in this case. So this is actually from the original paper where they looked at sort of viability of cells with point mutations, and you can see they cluster into basically two clusters, and all of the nonsense or sort of protein stop inducing mutations sit in this bottom cluster, and all of the kind of synonymous mutations sit in this upper cluster. But the missense ones, so the ones that we don't know the function of, they split between these two different clusters. And that means that you can then start classifying them based on whether they have an effect on protein function or not. And this is being done very large scale as part of this AEVE Alliance, the Atlas of Variant Effects Alliance. I'm not going to go into this through lacks of time, but I think I'll skip to this one, where so I've talked about some of the dual guide and SNP experiments that we're doing here. We're also looking at overexpression in these systems, and also using sort of more complex readouts. So everything from these kind of cell death readouts through single cell, and we're now branching out into these kind of imaging-based assays of cell function as well. So I think this is incredibly powerful sort of methodology. The other sort of things I'd like to just briefly go through are kind of other applications of this kind of method. So firstly, I've talked almost exclusively about Cas9. There are various different engineered variants of this Cas9 or Cas9s from alternative species. So these can actually massively improve your ability to do certain experiments or improve the outcomes. But there's also other technologies as well, as Shalini mentioned. The kind of Cas12 system is one that is being used quite a lot by people at the Broad. This is slightly different in that the CRISPR RNA is much shorter, and it also has array processing activity. So you can express sort of maybe 4 guides as an array, and it will process them itself into individual CRISPR RNAs. So you don't have to express them separately. And also allows you to kind of actually combine that with other systems because it's a completely orthologous one. The PAM sequence is a little bit more restrictive, so that does restrict some cases. And then one which you'll hear a bit more about later on in the week is also this Cas13 system, where instead of targeting DNA, it's also targeting, or it targets RNA. And that not only allows you to degrade at the RNA level, but it also allows you to also create a dead version of that and fuse it to RNA modifying enzymes that are quite powerful.
Andrew, the other Cas9, like SA-Cas9, are they better or equivalent to SP-Cas9?
No,
Or
is our experience. Again, we tried about, I don't know, a dozen different versions very early on. Some of them have advantages. So SA-Cas9 is shorter. So for therapeutic applications, it's beneficial because you can fit it into a lentiviral vector much more easily, or an AAV. Most of the others seem to be much more chromatin dependent. So SA-Cas9 and strep pyogenes Cas9 are very much quite independent of chromatin structure. And I think we're quite lucky to pick those as the starting ones, or they screen through an awful lot. Most of the others that we've tried, whilst they have different PAM requirements and various other sort of things, they are very much more dependent on chromatin structure. So we haven't had any success with most of those, apart from at the odd site where they work very well, but not generally. The engineered variants, much more so. So we use the enhanced specificity variants a lot, and we do use the engineered PAM variants, so ones with a slightly less restrictive PAM sequence. It should be mentioned they do have lower activity, definitely, but it's still present.
which one did you say is lower activity, the SA?
SA-Cas9 is probably on average slightly lower efficiency, but it's the best of the rest after strep pyogenes in our hand. Yeah.
The NG one, can we use it for like, is it good? Like, can we use it for the base editing if we are not getting the
Can,
SA?
yes. Again, it's less problematic. So for nuclease, it lowers its activity quite a lot, the
Okay.
NG version. But for base editing, it seems to be slightly less dependent on that. It also it depends very much on the system that you use as well. So if you're using a constitutively expressed sort of protein, it's much less dependent because you have a longer time to actually deal with or introduce the mutation. So if you're using RMP, it's much more dependent on the activity of the enzyme.
Andrew, I would also have a question, which is a bit related to Shalini's question. For now, the Cas12, because you said you tried that for the combinatorial screens, right? And the efficiency was not as well as with the Cas9. Did you then also try the enhanced Cas12, where you have like direct repeats and NLS?
Yes. Yeah, no, that's based on that.
Okay.
If you actually look at the papers, they never do a direct comparison of one guide with one guide. And it is lower efficiency with one guide and one guide. But you have to dig through them in quite a lot of depth to be able to see that. It's not present, if you like. So yeah. And if you talk to John, it's kind of he would agree with that, I think. And then I guess sort of finally, it's I think worth noting that actually you can make a couple of point mutations in the Cas9 protein that eliminate its nuclease activity, and this means that, but it still retains its DNA binding activity. So that means you then have basically what is a sequence-specific DNA binding factor that you can then fuse whatever activity you'd like to it. And this has been used in a lot of things, and I'm going to go relatively quickly through these. One of them is base editing, where you fuse it to a cytidine deaminase domain, and this actually deaminates cytidine, so it turns a C into a U, and then that, when during replication, ends up being converted into a T. And there's various different tricks you can do here to actually bias repair in the direction that you want it to do. Again, dig through the papers if you're interested. But there's also different classes of editor which are amazing sort of protein engineering exercises where you can now do certainly A to G editing and to some extent C to G editing as well now. And actually with Matt, which he may well talk about later, you can actually use this for screening across across sort of, in this case, a gene. So this means that you can do very similar screens to what I've talked about with knockouts. Again, you're reliant on the efficiency being high because you need the guide to be a proxy of the genotype, and therefore, if the efficiency is not high enough, that's difficult unless you sequence the region that you're interested in when it can get around that limitation. It is hard to interpret a negative because if there's no introduction of a mutation, it's difficult to know what that actually means. But again, we're kind of trying to we've been working out ways of doing genotyping and a functional readout on the same cells. So that means that we can get around that to some extent. Another one that people will probably have heard of is prime editing. This is actually where, again, an amazing sort of protein engineering exercise that I can't believe it actually works, where you have the guide targeting the genome, you have an extension on the guide that acts as a template for a reverse transcriptase that you fuse to your Cas9. And what happens here is actually the Cas9 is not a dead nuclease. It actually just makes a nick in one strand, and then the end of your PEG RNA, so the end of this extension, hybridizes to the genome, and then the reverse transcriptase extends the genome to include whatever edits you've introduced in your template. And then the whole thing is kind of resolved by various different endogenous DNA repair processes. That means that sort of, again, you can bias the repair in various different ways, and that's been actively working on at the moment to try and improve the efficiency of this, and in theory would allow you to do any base substitution in the genome pretty much, but it is highly variable in efficiency. So this is an example in HAP1 cells that we work in a bit, where we can get very, very high efficiencies, 85% efficiency at our favorite site. But it's very, very variable. So this can drop to virtually zero using different PEG RNAs. Similarly, in iPS cells, we can get sort of, in some cases, very efficient sort of editing. This is an example of about 15%. We've also got sort of others up to about sort of 60, 70%. But again, it drops to basically almost zero at some other sites. So you have to go through an awful lot of PEG RNAs in order to find one that is effective for your particular sort of mutation that you're trying to introduce. But the rules will get better as time goes on, I think.
Andrew, we noticed that the PEG RNA also gets integrated. Have you noticed that?
We haven't seen that too frequently, actually. Again, it's something that can happen, definitely. But yeah, we haven't seen that to be a massive issue with the ways that we're
For you?
hiding it. Yeah. This is pure sequence, if you like. This is the kind of level of pure sequence, this green one. And very recently, there's this paper looking at another way of doing something very similar, where you take your template to the site of the cut using this single-stranded DNA tethering domain fused to your Cas9, and then you have a DNA-dependent DNA polymerase rather than a RT to actually copy this template back into the genome. So this is, if you like, a kind of artificial way of being able to put that into the genome. And this, again, is lower efficiency at the moment, but I think there's a lot of scope for improvements in this kind of methodology. And finally, this was actually a figure I drew for a review a long time ago, and I think pretty much everything on this picture has now been applied. But you can fuse this to activation or repression domains, and these can be used to turn on and off genes. This has been applied in a lot of different sort of situations. I think probably skip over this one. One of the most recent interesting sort of ways of doing this is actually not just fusing it to a repression domain, but fusing it also to a DNA methyltransferase. And this means that then you can actually turn off gene expression, but also maintain the off state of gene expression, even without the presence of your initiating repressor, which is actually one of the issues with this, is that you have to maintain expression if you're just using a repressor domain. So you can see here that actually even after the expression of this construct has died off to virtually zero, you can actually maintain repression up to sort of, actually it's into the months or even years, which I find amazing.
In your experience, which repressor do you recommend?
So crab, the Zim3 crab is the standard one that everybody uses. The activation, I would say VPR is our sort of tend to go to one, but SAM can work as well, but it's a little bit more fiddly to get to work. We haven't done a huge amount with CRISPR-A or CRISPR-I, to be honest. That's kind of, but there's, again, it's something that's being actively developed at the moment. So there are better activators out there that are being tested. This one I think is an interesting one, but we haven't done a lot of testing about it. And yeah, these were some of the other things that we kind of thought about at that time. So you can use that DNA binding factor to bring in a GFP to look at the DNA loci, even in live cells. You can purify out bits of chromatin, recruit RNA to the RNA component here, enforce or break DNA loops, act as kind of a blocker to either polymerase or even transcription factors, or bring in chromatin modifying activities into that DNA. So really, it's limited by imagination, I think. So I think, yeah, I think I'll stop there, given I've sort of pretty much used up all the time. I think we're definitely getting crisper as time goes on. And just to thank everybody involved. So we have a core team here that does a lot of technology development, and then we have a number of people employed under Open Targets projects to use and apply some of these technologies to neurodegeneration. And I've talked about some of the work from many of the people on this slide. So thank you very much. I'm happy to take any questions.
Can I ask you one quickly? So you said like you have the Cas9 expressing cell line for screening. Can we buy them?
So those have mainly been made by Matthew's lab, and I'm sure they are available. Sort of
Is that IPS lines?
Work to do. No, there's not. We don't have very many IPS lines that I think we're happy with as a Cas9 expressing line, actually, because most of the time the issue is that during the differentiation process, the transgenes silence very effectively. That's true with safe harbor integration, with various different insulators, with piggyback, with lentivirus. So most of the time you're working with differentiated cells. So we have some lines that work quite well in IPS cells, but not really in differentiated cells very well. So we tend to deliver it in slightly different ways. So we deliver things as an all-in-one vector later on, as late in the differentiation process as we can.
I see. Thank you.
Thanks very much, Andrew. Yeah, it was great to see all of the real life data as well. Not just the, like the polished figures from a paper, but actually seeing the nuts and bolts of how things are working and the optimization that you've done. So,
Andrew, thanks for your talk. I was wondering about the Cas9 fuse with crab. Can you say lose-like activity after a while, just because, like, I think the transfection is transient, right?
Depends on how you do it. So often what people do is they make a stable line with Cas9 crab.
Like with the constitutive expression, right, of Cas9 crab. Yeah, I was wondering, why would you not like doing like that? Can you have, like, with a constitutive expression, like many other, I don't know, side effects or something?
No, I don't think so. Actually, surprisingly to me, Cas9 or dead Cas9s are not terribly toxic when you express them. Even if you overexpress in an entire mouse, they only get inherited at slightly less than Mendelian ratios. And it's no different to overexpressing a GFP, for instance. So I don't think there's any real toxicity. The issue we have, because we work a lot with IPS-derived cells, is this transgene silencing. And so during the differentiation process, generally the transgenes that express the dead Cas9 crab diminish over time. So there's fewer cells expressing them. And therefore, when you're working with a microglia, maybe only 30% of the cells will actually express that protein. And that means
even if it's constitutive, like inserted into the genome.
Yeah.
Okay.
Even if it is, it's to do with various different levels of repression. There's actually, if you're interested, there's a bioarchive paper that I'm on, but mainly is from Marius Vernig's lab, where they've developed a piggyback construct that avoids some of those. I wouldn't say it's completely perfect, but it kind of, by using optimized promoter regulatory element insulator sequences, we've managed to find a combination that works well. But it doesn't work in all cells. Microglia, it doesn't. For instance, it drops off in the middle of the differentiation, which is exactly where you want to actually get the editing to work. So, so actually, it depends what you're doing, but that's been a bit of a bugbear for some of those processes. It's doable in some cells, and in cancer cells, it's dead easy. It's not a big deal. But yeah, watch out if you're using IPS differentiation. I could give you a whole talk about the balancing.
Maybe I just want to know, do you keep selection on during the differentiation?
Yeah, unfortunately, we can't. In some cases, it depends on what it is. So we do when we can, but in many cases we can't because it disrupts the differentiation.
Differentiation, yeah.
Probably because actually you're killing off quite a large proportion of the cells in the dish, and that's a variable proportion. So it means that they then are not the right density to maintain the correct differentiation. I don't think it's something fundamental about the drug interfering with the differentiation. I think it's more to do with the cell densities.
Okay, thank you.